Today we will discuss symmetry and chemical composition of vertices. Only two kinds of symmetry have been recognized in vertices, icosahedral and helical symmetry. So first we will discuss icosahedral symmetry. The icosahedron is one of the five classical Platonic solids of geometry. Five classical Platonic solids of geometry include tetrahedron or pyramid, cube, octahedron, dodecahedron, and icosahedron. Icosahedron has 12 vertices and 20 faces. Vertices mean corners. It has axes of 2, 3, and 5-fold rotational symmetry. You can see here in this picture, 2-fold, 3-fold, and 5-fold rotational symmetry, passing through its edges, faces, and vertices. Icosahedron is the optimum solution to the problem of constructing from repeating subunits a strong structure to enclose maximum volume. Same principle were applied for construction of icosahedral buildings, geodesic domes, by an architect, Buckminster Fuller. Only certain arrangements of repeating subunits, these repeating morphological units like capsomers, can fit into these phases, edges, and vertices. For example, in the case of adenovirus, here capsomers on the faces and edges bond to 6 neighboring capsomers and are called hexamers. Similarly, those at the vertices bond to 5 neighbors are called pentamers. Next is helical symmetry. The nucleocapsids of several RNA vertices have helical symmetry. Here, the capsomers and nucleic acid molecules self-assemble as a helix or coil. You can see here in this picture. These are capsids and nucleic acid. Orange colored units are capsid made up of proteins, and green colored nucleic acid, DNA or RNA. And how the coil or helix is formed, you can see here in this picture, which is very clear. In all vertices, each capsomer consists of single polypeptide molecule. Plant vertices with helical nucleocapsids are rod-shaped and naked, means non-enveloped. Not any envelope is present on the capsid. However, in the case of animal vertices, helical nucleocapsids are wound into a coil and enclosed within a lipoprotein envelope, means envelope made up of lipids and proteins. Why this is so? Possibly to give nucleocapsid stability, to make the structure stable. Another type of symmetry can be found in some vertices, that is complex symmetry, where vertices are more complex, being composed of several separate capsomers with separate shape and symmetry. For example, bacteriophage. In this picture, you can see head and tail of bacteriophage. Head, symmetry of this head part is icosahedral and helical tail. Tail fibers are present and at the base, spikes are there. They don't have either icosahedral or helical symmetry, right? Due to complex structure. Another example of complex symmetry is a pox virus. Next is chemical composition. As we have already discussed in our previous lecture, there are mainly two components of infectious viral particles are there: nucleic acid and protein, right? In addition, all enveloped vertices contain lipid in envelope and carbohydrate in their glycoprotein peplomers. What are peplomers? I'll explain peplomers later. The largest and most complex viruses, for example, pox viruses, rhinoviruses, African swine fever virus, also have lipids associated with other parts of virion. Now we know that virus contain only one type of nucleic acid, either DNA or RNA, but never both. We have already discussed in structure of viruses. All viral genomes are haploid, means they contain only one copy of each gene, except retrovirus, where genome is diploid. Viral DNA or RNA can be double-stranded or single-stranded. So first we will discuss DNA. Genome of all DNA viruses consists of single molecule, which is double-stranded, except in the case of parvoviruses. Here, it may be linear or circular, right? Within the virion, circular DNA of pepoviruses like that of mitochondria and bacterial plasmid, it is supercoiled, circle, and called superhelix. In the case of DNA of hepatinoviruses, here one strand of circular DNA is shorter than the other. So for such kind of genome, we use the term partially double-stranded, not fully double-stranded. Then RNA, genome of RNA viruses may also be single-stranded or double-stranded. All viral RNAs are linear. However, in single-stranded RNAs of some viruses, for example, arenaviruses and vanyaviruses, have sticky ends. So due to the presence of these sticky ends, molecule can occur as a circle, right? And you can see here what are sticky ends. Here in this picture, example of single-stranded DNA, as we know that in the case of RNA, base uracil is present instead of thymine, right? So just in order to explain what are sticky ends. So sticky ends where base sequence are complementary to one another and enabling the two strands to be connected by base pairing. We know that 4 bases are present in nucleic acids. In DNA, guanine, thymine, cytosine, and adenine. Guanine can pair with cytosine, and adenine can pair with thymine. Thymine can pair with adenine, and cytosine can pair with guanine, right? So these bases are complementary to each other. In this way, a circle is formed. The genome of single-stranded viruses have unstable secondary structure, means a region of base pairing, probably serving as signals for controlling transcription, translation, or packaging into the capsid, right? Single-stranded viral nucleic acid, which is generally RNA, can also be defined according to its sense. It may be have positive sense or negative sense. If single-stranded RNA is of same sense as messenger RNA, it is called positive sense, means to have positive sense. For example, coronavirus, right? If its nucleotide sequence is complementary to that of messenger RNA, it is said to have negative sense. For example, in the case of arenaviruses, vanyaviruses, etc. Now, size of the single-stranded viral genome varies from 7.5 to 18 kilobase pair, and that of double-stranded RNA viruses from 7 to 22 kilobase pair. Right? Next, proteins. Virus-coded protein may be structured or non-structured. As it's clear from the name, structural proteins, they are part of structure of virus, virion, right? Non-structures, they are concerned with some other aspects like replication cycle. Major role of structural protein is to provide viral nucleic acid with a protective coat, that is capsid. Capsid is made up of protein, you know that. Virions of all viruses of vertebrates contain several different proteins ranging from 3 in the case of simplest viruses and to over 100 in the case of complex viruses, for example, pox viruses. Now, capsid proteins are assembled in virion in groups and to form capsomers, which are visible only in electron micrograms. Each capsomer is again composed of 1 to 6 molecules of polypeptide. When polypeptides are of same kind, they are known as homopolymers, but sometimes they are different, means heteropolymers. Other proteins like glycoproteins make up the peplomers projecting from the envelope. A second type of envelope protein is non-glycosylated matrix protein that occur as a layer at the inner surface of lipid envelope. In the case of paramyxoviruses, orthomyxoviruses, etc. Here in this picture, it's a diagram of coronavirus. You can see here these spikes made up of glycoproteins. These are peplomers. These brown spikes are there, projections are there. They are called peplomers, right? Another type means other type of proteins are E proteins, M proteins, or spike glycoproteins. So these are the different type of proteins present in coronavirus. Now, one or more of the proteins on the surface of virion has specific affinity for complementary receptors, which are present on the surface of susceptible cells. So next, lipids. Lipids constitute about 30 to 35% of the dry weight of enveloped virus. Viral envelope being derived from cellular lipids. So the composition of lipids of particular virus differs accordingly, according to the composition of particular composition of membrane lipid, right, of the cell in which they are present. Now, what type of the lipids are present in viruses? Phospholipids and cholesterol. About 50 to 60% of enveloped lipid is phospholipid, and the remainder is cholesterol, right? Pox viruses, rhinoviruses, and African swine fever virus, they contain cellular lipid in their envelope, and the other lipid in the inner part of virion. Similarly, lipid occurs in the outer membrane of pox viruses. They has different composition from that of the host cell lipids. Next, carbohydrates. Carbohydrates are mostly associated with viral nucleic acid. And apart from that, they occur as a component of viral glycoprotein also, which usually occur as peplomers. So, pox virus, they also contain internal glycoproteins in the membrane of the core, and one of the outer capsid proteins of rotaviruses, which is also glycosylated. So next, we will discuss viral envelope. Viral envelopes acquired at host cell membrane, right? Some at plasma membrane, other at internal cell membranes. Internal cell membranes like nuclear membrane, endoplasmic reticulum, and Golgi complex, right? Lipids of viral envelope are derived directly from the cell, but proteins in the envelope are virus coded, right? One kind of the glycoprotein, papillomer or spike. These papillomers can often be seen clearly in electron micrographs as projection, as already I have told you. So other proteins, enveloped proteins, matrix proteins, those proteins are non-glycosylated and found on inside the envelope of virions of several families, and they provide rigidity, means hardness to the structure. Right? Now, whatever we have discussed, on the basis of that, what type of the question can be asked in mains? As mains paper 1, section A and B, it is generally asked that describe the following in about 150 words, right? So, whatever we have studied, from that question, like chemical composition of virus can be asked, describe viral envelopes or kinds of symmetry in the virus, right? So whatever we have discussed in this lecture, material is more than sufficient to answer these kind of questions.